Thank you so much for inviting me to be a part of your meeting. I'll be talking to you today about the worldwide opportunities in clinical neuroimaging and what they can bring to studies of various disorders. My background, of course, is in psychiatric disorders. I primarily work in the neuroimaging of schizophrenia and bipolar disorder. And of course, the issue with these disorders is that we know they have a physiological underpinning, but we don't know exactly what it is or how best to assess it. We don't have quantifiable and robust diagnostic techniques. It can be very challenging to identify exactly what disorder a person has. And this is true in so many medical situations. And the presentation of the disorder can be highly variable. And there's some question as to whether the underlying physiology might give us a better understanding about the sources of variability in clinical presentations and in response to treatment, of course. So neuroimaging has a number of obvious advantages. It can be done in vivo. You don't have to wait for post-mortem analyses or doing tissue samples or anything like that. It tends not to have any side effects. If you are doing an MRI and you make sure the person doesn't have any contraindications, you're not filling them up with dangerous chemicals or anything like that. So it's a very safe procedure, and you can do it over and over and over again. You can have longitudinal studies with multiple kinds of scans, and the person can do this as often as needed. And of course, you have a variety of measurements that you can get from the same MRI scanner. You can measure the structure of the brain, the volume of white matter, gray matter, particular white matter tracts. You can look at the function of the brain in various ways. You can measure metabolic measurements in the brain to identify what's been happening in the brain and look for the metabolic markers. You can look at neuroinflammation in the brain. And of course, even more recently, you can now look at the concentration of dopaminergic neurons in the various parts of the brain in the fMRI without giving them any contrast. So there's all sorts of options in what you can measure, which gives you tremendous power in asking questions about the underpinnings of various disorders. But of course, there are also disadvantages in using neuroimaging like MRI. These are very subtle measurements. They're not very strong. They're not very obvious. They're also very indirect measurements. We're not directly measuring function or metabolism. We're measuring the after effects, if you will, or correlated measurements of these kinds of functions. Or these variables, these measurements can be highly variable in an individual from one day to the next, as can clinical symptoms in some cases. And there's also the issue of reproducibility. Because these are nuanced, subtle, and variable measurements, it is very easy for one lab to find one thing, and then a different lab, they find something else. They find the opposite, depending on who they're recruiting, how they're doing the analysis, how they're actually collecting their data and interpreting it. So the field as a whole is very aware of the challenges that come in reproducing results and actually finding common effects that we can count on. Over time, it takes a great deal of effort. So one of the leaders in developing these approaches towards reproducible neuroimaging is Dr. Paul Thompson from the University of Southern California. And back in 2010, he was already asking people, can we work together to do better in determining the large samples, to pulling together the large samples that we need, not through collecting data together and putting it all in one location, but taking advantage of the data that we are each collecting. If one person's collecting 50 subjects, another person's collecting 30, and another person can maybe collect 100 or 200, if you can actually aggregate the analyses, you can use each other's data to answer both the questions of reproducibility and consensus effects. What is it that we really see in worldwide studies of a particular question. And also reproducibility. How much variability do we see? What is it that brings about differences in results from one lab to another? So he started this in 2010. In 2012, they published their first imaging genetics paper. And by 2014, they were also already working with groups all around the world who were organizing around different disorders, focusing on schizophrenia, bipolar disorder, ADHD, major depression, etc. And this is now the status of the ENIGMA Consortium as of last December. So you can see there are participating institutions from 45 different countries and 500 medical centers worldwide, really on every continent. And they've been organized across different disease working groups. So there are groups that are contributing to schizophrenia, bipolar disorder, the psychiatric disorders, the neurological disorders, the other disorders, other groups. There are new groups showing up all the time. Epilepsy is actually one of the latest ones. Tourette syndrome is one of the latest ones. Any group that is focused on neuroimaging within a particular disease or disorder is absolutely free to develop another ENIGMA working group and make connections around the world and try to aggregate their approaches and try to identify really robust results. Besides the working groups, there are also worldwide efforts in genetics methods, genetics as applied to cortical volumes, subcortical volumes, white matter measurements, and developing methods for other genetic approaches. There are development groups looking at different imaging types. So besides the straightforward measurements of volume, perhaps they're also looking at hippocampal subregions, white matter issues, shape. How do we actually develop techniques that don't just work for the experts? They don't just work in one lab, but can actually be robustly applied across multiple groups and multiple institutions. Anybody who can collect imaging data can do these analyses. It's been incredibly successful. This was a paper, I think it came out in 2021. It says all the authors and their affiliations appear at the end of the paper because it went on for pages. The ENIGMA genetics of cortical thickness and surface area paper pulled together data from over 50,000 individuals from around the world, looking at thickness and surface area, and did genome-wide associations. And again, they didn't pull all the data into one place. Every site ran the same analysis, and then they could do a meta-analysis to say, where do we see consistent results across the genome for all of these different brain regions? They were able to actually tease apart several hypotheses supporting differences in genetic effects between surface area and thickness of gray matter, and looking at genetic pathways related to these measurements that also seem to be correlated with cognitive function, Parkinson's disease, depression, etc. So each of the ENIGMA disease working groups is organized by a few people. I together with Dr. Theo Van Erp, run the schizophrenia working group. Other people run major depression, bipolar disorder, alcohol use, epilepsy, etc. And one of the great things about the ENIGMA framework is because we're all agreeing to do the same analyses. In these initial analyses of subcortical and cortical thickness, we all did cases and versus control. So where do we see differences between individuals with schizophrenia and individuals without schizophrenia? And it's, with enough subjects, it's a global effect. So there are areas that are more affected versus less affected. And that pattern that we see in schizophrenia, where we're seeing strong decreases in temporal lobe, frontal lobe, is weakly echoed in bipolar disorder. They're not quite as severe as schizophrenia, but they can be very different in alcohol use disorder or major depression, where it's more middle temporal lobe than superior temporal or inferior temporal lobe. Very different in OCD, more global but not specific in attention deficit disorder. And autism has its own patterns. Epilepsy has a very different pattern. And 22q11, a genetic disorder which we all think is very related to schizophrenia and psychosis, actually shows a very different pattern where superior temporal gyrus is affected as it is in schizophrenia and bipolar disorder, but other parts of the brain show a very different pattern. And part of this international collaboration allows us to do comparisons across disorder in this way. So again, we're doing what we call prospective meta-analyses. Every group shares the results of their analyses. We agree to do a common analysis, but we don't necessarily have to share the data unless you're trying to do very specific analyses. The general pipeline is that everybody looks at their own T1s, they do curation, they check the quality of their images from the scanner, they run a standard software. FreeSurfer is freely available, gold standard segmentation software. You run standardized analysis scripts, and then we can actually do the meta-analysis to say across all of these groups who are doing a case control effect in the hippocampus, looking at hippocampus volume. Obviously, in schizophrenia, we find very, very strong effects in the hippocampus and very consistent effects. But even there, you can see that the estimated effect size can vary dramatically from one group to the other. And some groups would not publish their results as finding that the hippocampus was a significant effect in schizophrenia because for some reason in their group, they don't get a significant effect, but consistently across, when you look for the consensus effect, you do see the effect size is very consistent. And of course, we can do this for all of the subcortical regions and look at the sources of variability. Why in the putamen, where we see increases, generally speaking, if we look across the world, across different groups, we see that the putamen on average shows a weak positive effect in schizophrenia. And then you can say, well, it's actually due in some groups where you have a different duration of illness, you'll see a very different effect. If you have a different age of your subjects, you'll see a different effect. If you looked at the amygdala and you look at the proportion of males, you'll actually see that can have a consistent effect on your case control effect size. So we can actually tease apart why some groups see one thing and other groups see other things if we're all working together. And we see the same thing in the cortical analyses. We can across, again, now in the cortical effects within schizophrenia, we had many, many more samples working with us, many more institutions working with us, including data sets from within China, Japan, Australia, Korea, etc. We could actually identify this image that I showed you before, this pattern of cortical thinning in schizophrenia, but we can also look at the effects of controlling for the global effect of cortical thickness and identify that consistently the temporal lobe is more affected than you would expect on average, whereas the frontal cortex, where we know there is thinning, is really reflecting the average result, as is the posterior parts of the brain, whereas the motor cortex and parts of the parietal cortex are really less effective, relatively spared, even though they are statistically thinner in individuals with schizophrenia than they are in healthy controls. They're not as affected as you would expect by the average. And when you have 10,000 subjects involved, you really can get some consensus robustness, some sense of what is likely to be found and what is individual variability across groups. And then, of course, we can dig into more specific effects, start teasing apart the effects of obesity, because so many of our subjects are affected by metabolic syndromes and obesity issues. Well, how much of that is really driving our disease effects? What's going on with shape, not just thickness and volume, but actual shape and asymmetry issues? What are we seeing in early onset versus sort of standard onset psychosis? What do we see in relationship with symptoms, positive symptoms, negative symptoms? And we can extend across diagnoses as well. We can, of course, that we already talked about, doing cross-disorder comparisons across the psychiatric disorders and syndromes. We can look at clinical high risk for psychosis generically using the same framework and compare it to what we see in the full-blown diagnosis, disorder. We can combine data from the healthy subjects from all of these studies, some of which were looking at schizophrenia or bipolar, but for the healthy controls, throw it all together and we can actually look at what happens to cortical thickness thickness across the lifespan using ENIGMA techniques. We can compare substance abuse disorders to the psychiatric conditions and see where do we see differences or where do we see common underlying mechanisms, perhaps, for gray matter loss. And this was a recent paper, which was just phenomenal, looking at, again, the ENIGMA patterns of cortical thickness results in the 6 psychiatric disorders, schizophrenia, bipolar disorder, etc., and then looking at known genetic expression atlases to say, where do we see genetic expression driving cortical thickness, driving cortical areas, and how does that overlap with where we see the loss in the psychiatric disorders, and what does that tell us about the underlying genetic underpinnings of these disorders, the shared neurobiology of these underpinnings. Really, really clever approaches to looking at what does it mean that we're seeing these gray matter differences. Last year, in 2020, well, actually this year, but starting last year, we pulled together a 10-year retrospective in the Human Brain Mapping Journal. They had a special issue with 31 papers reviewing the ENIGMA efforts over the years and looking at technical developments over the past 10 years, clinical applications over the past 10 years, clinical integrations going beyond just here's what we see in schizophrenia, or here's what we see in bipolar disorder. What are the newer groups doing? What are the newer techniques doing? This is, again, the sort of the current layout of ENIGMA. There are different working groups organized by psychiatric disorders, neurobiological disorders, neurological disorders, genomics, sex and gender differences, organic data science, the informatics, the techniques. I want to say a little bit about the anxiety working group and the TBI working group, because those are disorders that, of course, they look at neuroimaging, and it's very clearly an umbrella term. You know, an anxiety disorder, there are many different kinds of anxiety disorders. It just as in schizophrenia, we know there are lots of different things going on in schizophrenia, but we don't have the clearly defined subtypes that they have in anxiety that they've been working on for so long, identifying different forms of anxiety. And part of what the ENIGMA anxiety group can do is say, what is it that is specific to anxiety, sort of common across all of these, and then what is specific to the different subtypes of anxiety. So they're taking a very explicitly hierarchical approach to disentangling the neurophysiology, and the traumatic brain injury people are doing the same thing. We have lots of different causes of traumatic brain injury. Do they lead to the same effects? How are these effects different? What can we actually say by working together, looking across these different groups? Really tremendous effects. All of these effects are available online. Anybody can go and pull out the results from these papers at the enigmaviewer.org. So you can download them, use them, and apply them yourself to other things. There are new ENIGMA working groups showing up all the time. There's no rules about who can start an ENIGMA working group. Paul Thompson is ENIGMA Central. He sort of helps organize things, very, very helpful. This is one of the newer ENIGMA working groups looking at neuromodulation, trying to get people who do deep brain stimulation, transcranial magnetic stimulation, direct cortical stimulation, the electrical stimulations, machine learning approaches, etc., ultrasound, getting all of these groups to start talking together, start working together, start applying ENIGMA techniques, these sort of shared meta-analytic techniques, to dig into these approaches more cleanly, more closely, to come up with standardization, discussions, harmonization techniques, etc. They are looking for people who are interested in joining. If you are doing this kind of neuroimaging, they're happy to have people join in. There's also software for automating these kinds of analyses. I won't say too much. Dr. Vince Calhoun and I have been long-term collaborators developing software that helps individuals do these kinds of analyses and share results while protecting your confidentiality and privacy of the investigators, or excuse me, of the participants in the study, so that you don't have to give up your data. And it extends the kinds of analyses that ENIGMA can do through doing decentralized analyses on your data. So in conclusion, I want to wrap up there. I know there's a lot to say here. Large scale collaborations, you well know, are excellent for understanding clinical heterogeneity, clinical variety, for getting robust results on representative samples. Similarly, large-scale neuroimaging collaborations are excellent for understanding robust brain relationships with disorders, with the variability in disorders, and for trying to disentangle the physiology underlying the various effects that we're seeing in the clinic. And methods and techniques are available to address and to be applied to many new disorders, effects, their causes, their prognosis, predicting outcomes, understanding the underlying physiological causes for mind, brain, gut, brain relationships, etc. So the psychosomatic medicine is just in an incredible position to take advantage of all of these neuroimaging techniques and collaborations. And I look forward to seeing it take off. I would be happy to have you contact me with any questions you might have. Thank you.